Board. Okay, so we are recording. Hello, everybody. Welcome, everybody. We are today hosting the first edition of the 2025 Journal Club series, hosted by my lab at the University of Rochester. And the first guest for this summer is Dr. Zheng Jiang from Baylor College of Medicine. Zheng has a BS in clinical medicine from China Medical University. He obtained in 2004, and then he went on to earn a PhD in integrative biology from Florida Atlantic University in 2009. He completed his postdoctoral training in neuroscience at Johns Hopkins University in 2015 under the mentorship of Dr. King Y. Yao, where he investigated the phototransduction mechanisms of intrinsically photosensitive retinal ganglion cells with a focus on melanopsin pathways. His research uncovered a novel phototransduction pathway in M2 and M4 iPRGCs, challenging established views on phototransduction motifs across species. In 2019, he joined the Department of Ophthalmology at Baylor College of Medicine in Houston, Texas, where he is right now an assistant professor. His lab's research focuses on melanopsin transduction mechanisms and the electrophysiological properties of retinal ganglion cells using high throughput methods. Zheng is we met not that long ago during ARVO in Salt Lake City. Your poster was amazing. It was this very intricate way of looking at how patterns of electrical activity across many ganglion cells can tell us something about who these cells are, basically. And so the title of the lecture would be Decoding Retinal Output, Electrophysiological Signatures of Ganglion Cell Subtypes in the Mouse Retina. Thank you, Zheng, so much for coming and for being our guest today and for sharing with us your great work. So I'm going to stop sharing this here so you can go ahead and put your presentation.
Okay. All right.
Awesome.
Okay, you can see my screen. Okay,
yes,
all right.
Perfect.
Yeah. First of all, thank you, Michael. Thank you, Mike. That's for inviting me to give this talk. I'm very excited to discuss my work with you guys. So in today's talk, I will show you some unpublished data from my lab, and I will also show you that a high throughput method that we developed to study retinal ganglion cells. So this method allows us to profile the electrophysiological properties of retinal ganglion cells at a large scale. And we have also collected comprehensive data from about 10,000 retinal ganglion cells. And we classified these retinal ganglion cells into 34 different subtypes. And in the second part of my talk, I will show you that using this method, we found a significant functional variation of RGCs in the 4 retinal quadrants. So first, let me give you a brief introduction of the retinal circuitry and start from here. So in the retina, photoreceptor cells are the light sensing cells. When they are activated by light, they pass the information through synapses to second-order neuron bipolar cells, and in turn, bipolar cells pass this information to the output neuron, the retinal ganglion cells. And within the retinal circuitry, there are also inhibitory interneurons provide lateral inhibitions to facilitate the information processing. So I want to emphasize here is that at the photoreceptor layer, that's from the point of view of what kind of data they have, is they have the data for like the pixel by pixel data. But when it's processed by the retinal circuitry, when it comes to retinal ganglion cell layer, so the visual information has been extracted. And it is generally believed that each subtype of retinal ganglion cells extract one type of visual information, such as color, edge, moving bars, overall intensity, so on and so forth. So in today's talk, we are going to focus on retinal ganglion cells. So the retinal ganglion cells can be classified into 46 different subtypes based on single-cell RNA sequencing technique. So by mRNA profiling, This is Nick Chen and George Sin's work. They can clearly classify them into 46 subtypes. And by their physiological responses, by far, the most comprehensive classification has been done by Euler's lab in 2016. So they record the retinal ganglion cells using calcium imaging, and they gave a variety of light stimulation patterns. And then according to the response of ganglion cells, they can classify them into about 32 different RGC subtypes. And in my lab, we are using electrophysiological properties to classify retinal ganglion cells. The reason to use electrophysiological properties for three major reasons. One is by doing electrophysiological recordings, you have very high temporal resolution. So you can come to 20 kilohertz data acquisition rate. In contrast, when you are doing calcium imaging, so usually the acquisition rate is about 10 hertz. So this is about 20,000 times higher than the conventional than the calcium imaging approach. And also while we are recording retinal ganglion cells, we can directly record the action potential firing. That is exactly what the retina tells the brain. So this is very so the action potential firing is much more accurate than the calcium change of the cell, and it directly represents the output of the retina. And the third is while we are doing electrophysiological recording, we do not need light for the data acquisition. So in contrast, for calcium imaging, so while you are measuring the calcium concentration, usually you need visible light. Even if you are using 2-photon calcium imaging method, there is still a chance to activate rhodopsins and activate retinal circuitry. So this allows us to study the retinal behaviors in a scotopic light intensity range. And for people who are not that familiar with electrophysiology, let me give a brief overview of the methods, the traditional method. So one is called wholesale recording method. So this has been used widely in the field for more than 4 decades. So basically, you need a sharp glass electrode with opening about one micron to approach the cell. And then you can form gigasil onto the cell and then record the light response from the cell. And meanwhile, you also can put dye into your electrode. You can see the morphology of your cell at the end of your recording. So this method provides very good data. So it gives you really high signal-to-noise ratio. And in addition, you also can clamp the cell at different voltage to control the response, to control the cell. And also you can look at the inhibitory inputs, excitatory inputs. The only disadvantage for this method is this is not a high throughput method. So you only can record one cell at a time. So usually experienced postdoc probably can record 5 to 10 cells each day. And another somehow more high throughput method is called multi-electrode array, also called MEA. So for the conventional MEA, so usually they have this is about 60 electrodes, 64 electrodes to be precise. And then you place a retina on top of it. This is essentially an extracellular recording method. And you can usually, in one setting, I believe usually you can do about 20 cells in a good prep. And disadvantage is that you only record tens of cells, and the distance between each electrode is really large. It's about 200 microns. So you are sampling the retinal cells in a very sparse array. And in my lab, we are using a new technique called high-density MEA recording system. So here you see this is a head stage of the high-density MEA. And if you zoom in, this is a recording chamber. And if you further zoom in, you can see this black area is the sensor area. This is about this is exactly 1 square millimeter. Within this 1 square millimeter, it's packed in with 4,000 electrodes. And then you see this white tissue on top of it. This is a piece of ex vivo retina. And so we can perform recording of this retina. So during each recording, we usually can get about 200 cells from this area. And then and if you do the math, you can calculate that's according to the denser has a relatively high density. So between each electrode, the interval is about 16 micron. And here you are doing a recording.
Zheng,
Yes,
sorry to cut you off.
sure,
So
anytime.
the interval between each electrode is 16 microns, and how big is the electrode itself? Is another 16 microns or
So
less?
this is about 8 microns,
Eight.
roughly about
Okay.
8 microns, half of the size. Yeah.
And the electrode is a little square shape or is a circle?
So it's a circle shape.
It's a
And
cylinder. Okay.
yeah, it's a circle shape. And then it's interesting that you ask this question. So you can see it's a circle shape, and then in between each electrode, there are gaps. So within these gaps, actually, so the chip, the manufacturer, they put more electrodes outside, so in the gap area. So in the gap area, that's actually you can control the voltage in the gap area. Basically, you can electrically activate certain part of the retina and then record the interactions. So that part we haven't explored yet. So currently we just use the light for stimulation, but potentially this system allows you to do the electrical stimulations. Yeah. Okay. And then for this one, that's you can see that's with 4,000 electrodes, and then you are doing data acquisition at 20 kilohertz acquisition rate. So this produces a lot of data. So the data size is 10 gigabytes per minute. So each day, one of my postdocs produce 2 terabytes data. So, and so far we have collected 300 terabytes data in the lab for retinal ganglion cells. And then this is to store this data, do the spike sorting. It's not trivial. So literally, I built up a miniature data center in the lab. We have 9 computers running 24/7 to process this data, and we also have 3 file servers to kind of store this data and back up this data. Uh, and then during the recording, let me describe the procedure in more detail. That's we first sacrifice the animal, take out the retina. So within a few hours, that's how the retinal ganglion cells are still healthy and light responsive. So we put the retina on top of the chip with retinal ganglion cell layer facing toward the chip. And then the entire chip is placed under the microscope. So we hijacked the light path of the microscope, and we placed a OLED display in the light path, allows us to project any video or images on top of the recording area. So in this way, we can program whatever the video we want to do. So from full field light stimulation to moving bars, white noise stimulation, we even have natural scene stimulation. Basically, you're just kind of play a video on top of the retina. And then because these videos are natural scenes, so you have a lot of the kind of natural visual features that you can extract. Yeah, but for today's data, that's mostly we are focusing on this pattern image. So we give the retina 6 different stimulation patterns. So I will describe them one by one. So first, it's the simplest one. It's just a step up. So basically, it's just the full field white light from dark to white. And then while you give this stimulation, you will get a light response from each ganglion cell. This is showing one ganglion cell. And then y-axis is the firing rate. And you can see it has this should be an unsustained ganglion cell. you see it have a transient peak followed by a sustained response, and then at the off, it goes back to baseline. So simply look at this trace, we can extract a lot of numerical features. For example, we can get the on-max value describe this transient peak. We can have unsustained value, which is average at the sustained period. And if this is off response, we also can get off max, off sustained as well. We also can get the baseline mean, baseline standard deviation. And in addition, we also can engineer some feature. For example, that's how we can calculate the ratio of the on max to the unsustained. Then we have a transient to sustained ratio. And we also can have on-off ratio if this is an on-off cell. So by doing this analysis, we get about 13 different features simply from one trace. And furthermore, that's the next stimulation we gave is green and blue light stimulation. So in this, similarly, we also can extract lots of features from these responses. We also can get green to blue ratios as well. And the next is we call it frequency step. So basically, this is full field light stimulation. The light goes on and off at different frequency, from 0.5 hertz to 5 hertz. And you can see this cell is following it at low frequency very well, and it's not following it that well when it's reaching to the high frequency. So this provides some information about the temporal resolution of the recorded cell. So again, that we also can extract many features like the amplitude, phase shift of each frequency. And next, we gave a stimulation pattern. This is a moving bar stimulation, which is a horizontal bar moving across the receptive field of the cell. And then we can analyze that whether this cell has any direction selectivity. So this cell do not have direction selectivity. So you see it's responding to the moving bar at all directions almost equally. And if it's a direction selective cells, then we can extract the direction selective index to describe, kind of numerically describe how good, how strong the direction selectivity is. And furthermore, we also do some more systematic stimulation pattern. So this is called a spike-triggered average. So this is basically what we do is that we give white noise checkerboard stimulation. So refreshing rate is at 30 hertz. So basically, you are playing some randomly alternating black and white checkerboard stimulations. And then, meanwhile, you're doing the recording of all the ganglion cells. And then after you finish this, you get a data that's of what's the video you have played and when the spike has been triggered. And then once you get this data, you can come back to ask, before each spike, what kind of video the cell have seen. And then if you have 3,000 spikes, you will have 3,000 videos for you to analyze. So here, actually, this is relatively a simple analysis. Is you just average all the videos before every spikes. And then once you average them, the part that is randomly alternating will cancel each other. And if you have a hot spot, this hot spot will be highlighted. By doing this, you will be able to get a receptive field of the cell you recorded, and you also get other analysis and other parameters to describe the cell features. So for here, we can get the spike-triggered average in 2D. Basically, it's a receptive field. We also can get the kernel, the kernel of the spike-triggered average. Furthermore, we also can do linear and nonlinear fit to describe how well, so kind of the linear properties of these ganglion cells. So in this way, we also get 5 features out of it. Yeah.
Sorry, Zheng. I'm sorry. We have 2 questions.
Sure.
The first from Soma is, what is the pixel pitch of the OLED?
What's the pixel pitch?
Of the OLED.
Of the OLED. So the The hardware we have provide 800 times 600 pixel resolution. And then, but here, if you are doing the spike triggered average, so here each square, so this I think, so we have it. This is a 15. Well, we have is 15 times 15 checkerboard. So I cannot do the calculation right now. So, but it's basically it's 600 divided by 15. So this is 40 pixels for each cell over here.
Okay.
Yeah.
Nice.
Mm.
Second question from Sabina. Here,
Mm.
where actually what you're showing right now, the STA analysis, how long before the spike onset time is usually analyzed for STA? I guess she's asking how long you take it from the spike backwards.
Oh, so that one you can digitally operate that. Is if you want, you could do it as long as you could, as long as you have the as long as you have the computational power. But for usually that's for the wild type retina. 1 second is long enough. And you can extend it to longer time, but usually that's if you extend it to longer time. I have tried that. I've played that with even like 20 seconds. You don't see anything. So beyond 1 second. So usually you just do the analysis for 1 second. Yeah. Mm.
Awesome. Thank you.
Yeah. Okay. Sure.
Those were the two questions for now. Thank you.
Yeah. Okay. All right. Mm. Okay, and then by doing this, we can get the kernel of the response and linear fit. And after we finish all these 5 stimulation patterns, then we add synaptic blockers into the solution. So the reason to do that is we want to identify intrinsically photosensitive retinal ganglion cell. So these cells, they do not need rhodochrome photoreceptors to provide light responses. What they have is that they have a melanopsin to be expressed intrinsically. So even without circuitry inputs, they still can respond to light. And this is not an IPRGC, so you do not see light response. But if it's IPRGC, you will see a response here. So altogether, these are 6 different visual stimulations and gives us a lot of numerical features we can analyze. And furthermore, so in the retinal ganglion cell layers, there are retinal ganglion cells. There are also displaced amacrine cells. So to differentiate these 2 cell type, we use this is also a spike triggered average performed on the field potential. So basically, you put all the electrode voltage through the spike triggered average. In this way, you can visualize the propagation of action potentials. And here you are seeing is this retinal ganglion cell action potential initiated and propagate along the axon. So this is slowed down by about 1,000 times. So what you see here is it happens in a few milliseconds. So you only can see this with electrophysiological recording. You won't be able to see it with calcium imaging. This is too fast. So, and then in contrast, so some of the cells, they do not show a action potential propagation. So then they must be a displaced amacrine cells. So in our data set, we automatically identify these retinal ganglion cells using our customized algorithm. And then all the cells we included in our database are retinal ganglion cells. And altogether with all these features, and then we also cleaned up the data sets by excluding the displaced amacrine cells. We get about 10,000 retinal ganglion cells in this data set. And then, so each of them has 39 different visual features to describe their light responses. So here you see this is a quite comprehensive description of retinal ganglion cells. And then every cell, I can see that we know a lot about every cell we recorded. And if you visualize this data, here you are seeing about 3,000 cells. So each row is 1 cell, each column is 1 feature. And then if you are, all these features are numerical features. And if you look at this data, so you probably will think that this looks like single-cell RNA-seq result. That's except that's these are not the mRNA expression profile. These are the electrophysiological property profiles. And then, but for given this data set, that's um, uh, it looks so similar to the single-cell RNA-seq data set. It's all high-dimensional data. Each cell is represented by a high-dimensional vector. And then we can use the algorithm that the single-cell RNA-seq field have developed to analyze this data. For example, we can cluster them. We can ask the first question is, how many subtypes are there? And then we tried many methods. So far, this is a method that works the best. So first, for all these retinal ganglion cells, we separate them into three major groups. Based on their direction selectivity, that's we can identify all the DSGCs. These are direction selective retinal ganglion cells. And given on their intrinsic light responses, we can select all the RPRGCs. And the remaining cells, we put them in one group called other RGCs. And within each group, then we can perform unsupervised clustering method. So here is using UMAP and the dbSCAN method. So we can cluster other RGCs into 19 different subtypes. And we can do this to all the other subtypes, all the other groups as well. So altogether, we get 34 subtypes of retinal ganglion cells. And next, we can further visualize the data just to confirm ourselves we are picking up the right cells. So basically here you are looking at the other RGC groups. So each plot is one subtype, one cluster, or you can also call it one cluster. And the red curve indicates the average response of all the cells in this cluster. And then the transparent trace in the background indicates the individual cells response. So here I'm only showing you the step light responses. And by looking at this, we can kind of have some visual confirmation that all these cells we picked up, they have similar responses. And we can also do this to the SGCs and to confirm that the clustering are making sense. And for RPRGCs, here I'm showing you their intrinsic light response. So we can see there in the presence of synaptic blockers, that's these cells are still respond to light. And some of them have much faster response. Some of them have much slower response. And this faster response, they might be the M1 RPRGCs, and the rest might be M2 to M6 RPRGCs. Here we see 7 different clusters. So in the field, it has been reported there are 6 different subtypes of RPRGCs. So possibly these two clusters has faster response. Maybe they all belong to M1. And M1 RPRGCs is known to have BRN3B positive and BRN3B negative M1 RPRGCs. So possibly these are the subtypes of M1 RPRGCs. But so far, we don't have other evidence to support this. This is just a guess. Given our algorithm, we have these 7 different clusters. Okay, so with all this, we are able to classify retinal ganglion cells into 34 different subtypes, and we confirmed by visualize their responses to confirm that they are genuinely have similar light responses. And then we can ask.
Sorry,
Sure.
sorry. Before you move to the next thing,
Yeah, sure.
there is a question about the IPRGCs. Are you still using the same set of stimuli for IPRGCs or any specific wavelength?
So for the intrinsic light responses stimulation, so this is using a very bright light, bright blue light, which is about 100 times brighter than previously used for the normal stimulation. So it's coming from a different light source. As the RPRGCs, they require much higher light intensity to activate. And also, if you want to see the M4, M5 RPRGCs, they have much lower melanopsin density. So you do need much higher light intensity. So I'll put another way, this is like the 5 to 10-fold brighter than the direct sunlight intensity. Yeah, okay, all right. Okay, and then with these tools in our hands, then we can ask scientific questions. So first is, so here is that's the first question we want to ask is, do the physiological properties of RGCs vary in the foreretinal quadrant? So why I'm interested in this question? So one reason is in the literature that it has been reported that so the distribution of our RGCs are not consistent across the foreretinal quadrants. And but lots of people believe that visual information is processed by similar parallel circuitries in the four retinal quadrants. But the morphology data showing there are some outliers of the retinal subtypes. For example, this is Richard Wang's work. Her lab have studied one type of alpha RGCs and showing that this subtype of alpha RGCs has much higher density in the temporal region and much lower density in the nasal region. And then if you zoom in, you can see that's for the temporal region, that's you have higher cell density and a smaller receptive field. And in contrast, nasal region is larger and sparser. So this suggests that some RGCs may not evenly distributed across the retina, and then they also may implant different processing algorithm. But this is just for one subtype of RGCs. So a more comprehensive study has been done by Chen's lab. So they have used the spatial transcriptomics techniques to systematically study the distribution of all RGC subtypes. These are based on the mRNA profiling. And they found that among 45 different RGC subtypes, only 3 of them shows differential distributions across the 4 quadrants. So this is interesting, but only a kind of a minority of them that's showing a variation. So, but this study is based on the distribution of the cell soma. Whether the cells are still performing, whether the physiological properties of retinal ganglion cells are consistent across the 4 quadrants. So far, no one is able to answer it systematically. And here now we have our technique to record the retinal ganglion cell at a large scale, and we can address this question. Actually, the answer of these questions is all reside in this data set. So here we have 10,000 cells. We know the subtype of them. And even better, that's when we are recording these cells, we annotated which quadrant we recorded the cells from. And then simply by data analysis, we can split these cells into 4 quadrants and analyze the distribution of these cells in each quadrant. We also can analyze the electrophysiological features of these cells in the 4 quadrants. So here is take example for these other RGCs. And then we can split them into the 4 quadrants. Here, for example, you can see that these green cells have these clusters. It looks they are quite similar across the 4 quadrants. But in contrast, if you look at the orange cells, it seems that there is some difference in the ventral quadrants. So we can do this to all the cell types that's in each groups that we classify. So here, it just gives you an overview of how much variance you are looking at for the physiological properties across the 4 quadrants. The more detailed analysis should be done by looking at one cell type at a time and one feature at a time. For example, so this is one cell type, cluster number 7. This looks like a off cell. This is a step light response. And then we can look at the distribution of this subtype. And we also can compare the unsustained value. Basically, it's measure of the average of the sustained period. And we see that unsustained value across the four quadrants do not significantly change. And we also can look at the off max value. But the off max value is, the statistics shows that they are significantly different. And then the nasal and the temporal part is significantly different to the others. So here you are looking at only one cell type and two features. So given our data set, and what we should do is we should do all the 34 different cell types and all the 39 features. And so here we're talking about 1,000 ANOVA tests. And this is what we have done. So this is this plot. And so here is every tiny, excuse me for the small plot because there are so many features to show here. So each row is one cell type. Each column is one feature. Whenever you see the background turned red, it means the ANOVA test suggests the p-value is showing is a significant difference across the four quadrants. And then, but here, if you look at this, it looks like there is many features, shows variations across the four quadrants. And if you look at the cell types, at least have one feature is different across the four quadrants. And then you will end up with about 70% of the ganglion cells shows some variations across the four quadrants. And this is kind of, to me, this is quite surprising. It suggests that across the four retinal quadrants, that the visual circuits are quite different. And then this is against that we previously believed that the retinal circuitries are processing informations in a parallel processing algorithm. And the retinal circuitry are more complex than we previously thought. And one more thing to mention here, so this is more of a kind of a statistic issue. So here, what we are looking at is we are looking at lots of ANOVA tests and t-tests. But every time when we're doing the t-test, we have or ANOVA test, we set the significance value at p equal to 0.5. That is, we allow 5% chance for us to be wrong. But if you are doing the test for 1,000 times, so the chance for you to be wrong is very, very large. So here, in order to do this test, we have done a mathematical correction, which is called Bonferroni correction. So basically, we set up the p-value really low. So here, the p-value is set at 10 to the power of minus 5. And whenever you see a difference, these are significant difference. So we are not doing anything like the p-value hacking here. So altogether, I have shown you that we have created a data set of various light responses from about 10,000 retinal ganglion cells. And we did a unsupervised clustering and classification method. We found there are 34 different RGC subtypes. And we also found that majority of RGC subtypes have significant functional variation in the four retinal quadrants. And then it also demonstrates, so this is also a demonstration that potentially this can also be used to study retinal diseases. Currently, I have collaboration with other disease research labs that say, for example, you are asking for in glaucoma retinas, how does the retinal ganglion cell subtypes change their behaviors? So this is very powerful tools and very sensitive tools for to evaluate the retinal ganglion cell physiological change. And at last, I will say, I will tell you some ongoing experiments that we are currently doing. This is a very fun project, as I would love to share with you guys. So given our data set and given this method, we are trying to develop a way that we can predict the retinal ganglion cell firing patterns using machine learning. So here, if you think about it, with high density MEA recording setup, We have very precise control of the input, which is a light stimulation patterns. We can program it to any patterns we want. And we also have very precise recording of the output, which is a retinal ganglion cell firing. And then that's in between, we can treat it like a black box. So you have the input, you have the output. Basically, from the people who are doing AI, they are very happy to see this. You have a training set, very well-defined input, very well-defined output. And even better, we can classify the ganglion cell into different subtypes. And then we also can analyze the data in a subtype specific manner. And then to do this, that's how we build a currently we are still developing this. So we build a simple machine learning algorithm, which is a 3-layer of CNN neural network combined with 2 dense layer of neural network. And then we just the data set, we create a new data set, which is we just showing the retina in different pictures. So these are arbitrary pictures coming from a well-known, that's a computer vision data set. So these are some image from Instagram, random pictures like animals, people's, kind of cars. So we randomly pick up 1,000 images to present them to the retina. And then we present them for half seconds, and then the next half second is gray screen. And we keep on doing this for about 20 minutes. Within 20 minutes, the retina have been seeing 1,000 images. Meanwhile, we recorded their light responses. And then we can build up our training set. So basically, it's the input is the image, output is the firing rate patterns. And then after we do that, we feed the training set into this machine learning architecture, and we can train a model to predict how the retinal ganglion cell responds to an image it has never seen before. And then here is the test of the image of the result. So the blue curve is the response that we actually recorded from the cell. And the red curve is the response we predicted. And you can see from most of the response. So x-axis is time, y-axis is relative firing rate. And you can see that for most of the cell, we can predict the firing rate pretty well. But still, there are about 10% of the cells we fail. Kind of this goes crazy. It's kind of the opposite of the actual response. And then, but here, this is the idea. So we want to build an in silico model that's using sophisticated machine learning or AI algorithm to capture the retinal ganglion cells algorithm. And if we have this model for each subtype, and then we can do a lot of kind of in silico study, theoretical study on the retinal ganglion cells. And hopefully in the future, that's if you ask me, that's if I play this video or present this image to the retina, how all these subtype of retinal ganglion cells going to respond to that. So that's the ultimate goal. But currently we are in collaboration with a bioinformatics lab, and they are making more sophisticated algorithms to refine these models. And hopefully, so that's in the future, I can tell you a more exciting story.
Yeah. There is a question.
Yeah.
The question is, do you recurrently run the model to predict the response at each time step?
Predict the response at each time step.
Do you recurrently run the model? I think he's asking maybe, do you run this every time? The person that asked you, how would you want to unmute yourself? And yeah, go ahead.
Yeah, so I was wondering because your model is outputting one, so the x-axis has 9 different time steps. So the model simultaneously outputting the 9 responses at once, or every time you invoke the model, it produces output at a particular time step.
I see. Yeah.
And if so, are different invocations independent of each other? But obviously the responses are not independent of each other over timestamps, right?
Yeah, they are not independent of each other. And so while we are predicting them, so all these 9 steps are predicted at once. It's not kind of recursive predicting approach. We just currently we're just trying to go as simple as possible. So see, for example, this long short-term memory model architecture supposed to predict it even better. And we are working on to refine these models to try different methods to get better predictions. But for now, what the data you have seen, they all predict at once. All right, okay. All right, and so this is still ongoing. That's hopefully we can. So the ultimate goal is that we can have maybe another 10,000 retinal ganglion cells, and then with all this data set, with the recording from arbitrary images has been documented in a data set, and then we can train a model that can accurately predict ganglion cell output.
Right.
Okay, and this also has more significance in computer vision. So if you think about it, this is so for people who are doing the object recognition in the computer vision field, they are trying to design an algorithm that can extract useful visual features and then to do the analysis. But actually, in the biology system, that's in the retina. So there has been hundreds of millions of years evolution to create a built-in circuitry, a very well optimized algorithm just there within the retinal circuitry. So the goal is to extract this information, extract this algorithm that can also, so if we can use that for the computer vision, it can become a big boost. That's for the field as well.
Yeah.
Okay.
It sounds amazing.
Yeah.
Thanks. Please go ahead.
Yeah. And that's the last slide. And at last, I want to thank people who has helped us. That's Dr. Samuel Wu from our department and Ben Frankfurt. We have collaborated on the glaucoma research. And Wei also working on the disease models. And Junxin Wang, which is he is an expert in bioinformatics, and then we are collaborating to implement more sophisticated AI algorithms for the model training. And also Jason Chen and my collaborator at Johns Hopkins. And these are the people in the lab. And Dr. Yan Zhu, who has been doing most of the recording work. So basically, she single-handedly collected this 10,000 retinal ganglion cells. Yeah.
Awesome. Amazing work, really amazing work. Thank you so much for showing us this today. Thank you for being with us. I have a few questions, but here. There is another comment here. It would also be interesting to see if the artificial network gives us similar receptive fields using STA to the measured ones. Once the network is able to predict the spike trains. Have you tried this yet?
Can you see the question again?
So it would also be interesting to see if the artificial network gives us similar receptive fields using STA to the measured ones, the ones in the real retinas. Once the network is able to predict the spike trains. Have you tried to do this yet? Got it.
Okay. All right. So we have tried to use that to predict the using the white noise stimulation to do the prediction. But however, it's not very successful. I think the reason is that for the white noise stimulations, you do not have enough natural features. Things are quite random. And then, so we can, to a certain degree, we are able to predict, but so is there kind of a ceiling of the accuracy we cannot pass through? We only can get maybe 60% of the accuracy that's for the prediction. Never as good as this kind of image simulation. I think that is, my interpretation is that for ganglion cells, they are evolved to extract certain useful visual features. And these visual features exist in the natural scene images. And then white noise is just, if you think about it, of how many possible patterns a 15 times 15 grid can give you. That's a gigantic number. It's impossible to iterate all these possibilities. And then we are only sampling a very, very small fraction of the possible patterns. So we have to use something with kind of a meaningful visual features to make the training work. That's I think it's in that way doing things using random pattern is not that helpful.
Similar to previous question, do the early layers of the network resemble spatiotemporal receptive fields of the actual cells? What do you mean with early layers? Soma, can you
Yeah, I'm just wondering, in this model that you constructed, um, you have several convolutional layers that I'm not sure how much they could resemble like the linear transformation of input to spiking activity, but like have you looked at any of these layers to see if, for example, Conv1 or Conv2 have center surround or anything resembling actual neurons?
Yeah, very good question. So is this interpretable? Is this interpretable?
Yeah, exactly.
Can I say COV1 are ganglions, are bipolar cells? So we haven't tried yet, but potentially we could do that. We could build a interpretable model that's say for example that's uh, use the numbers kind of exactly representing how many subtypes of bipolar cells are there, how many subtypes of hemicrine cells are there, to make it more resembling the real retinal circuitry. But I think the issue is, even if it's interpretable, it's uh, uh, it's more of a hand waving. Even if I can tell you that's maybe one layer representing a bipolar cells, and then it looks kind of the receptive field looks like a certain type of bipolar cells. The feature looks like bipolar cells. This is still somehow hand waving. It could be done, but I would not say that's very kind of helpful for us to understand the retinal circuitry. What I believe is if you want to dissect this layer by layer, the best way is kind of you go back, uh, find a method, maybe calcium imaging, to do the same thing for the bipolar cells, to do the same thing for the hemicrine cells, for the horizontal cells. And then you have the real ground truth data to build the previous network. But for now, this is not our ultimate goal. And if you think about it, that's actually the
hello?
contribute to the processing. Also, kind of for the synaptic connections. So there are many structures of the synapses. So they also provide feedbacks for the circuit for the algorithm. So all this added up to the complexity. So what I believe is eventually we probably need something more complicated than this simple neural network to really represent the ganglion cell algorithm.
I see. Thank you.
Sure.
Awesome. I think there were very good questions from mostly young trainees here, postdocs and PhD students, which I'm really happy to see joining the call. So thank you very much for everybody that participated. Thank you, Zheng, so much for the lecture. This will be available on YouTube for people to watch again and revise with your permission. So thank you very much, and we really look forward to interact more in the future and have more of your data coming out to the world, and especially your methods, because I think that the analysis that you're doing is just amazing and it's so elegantly streamlined, let's say. It's really, really amazing. Okay, so thank you very much. Thank you everybody for joining the call, and we will continue with the tradition of the Summer Journal Club in August with a second guest then. Bye-bye. Thank you, everybody.
Thank you so
Thank you.
much. Thank you,
Bye.
everyone.